Hi, my name is Reagan, and for my biology role model project, I chose Jennifer Doudna. Jennifer was born on February 19, 1964, and grew up in Hawaii, where she became fascinated with exotic plants and species. Doudna's parents influenced her interest in science by supplying her with science-focused books and visits to museums, and encouraged her participation in hands-on science experiences. Additionally, after reading about James Watson and Francis Crick's discovery of DNA structure, she became hooked on science. She also became interested in science after genuinely enjoying her chemistry and biology courses in high school and college. Doudna first studied chemistry at Pomona College, where she also participated in undergraduate research. She then furthered her education in the biochemistry field at Harvard University and received her doctorate. It was during this time that her love of RNA began to emerge. Doudna says an obstacle she had to overcome and still faces today is gender bias against women. She states that in her early years she didn't really recognize or look for signs of gender bias. However, as she has progressed into higher levels of science, she says she recognizes it more often. Doudna says she has noticed more gender bias in areas of higher levels of public policy and the government. In her free time, Doudna enjoys gardening and reading various books. This includes not only science books, but also other non-related novels. I admire how inspiring and genuine this scientist is. She encourages people who have an interest or passion to embrace that passion and go for it. She states, "The most successful of them are people who are able to deal with their fears. We all have fears, but sometimes you try something and there is failure, right? You have to deal with that. It is easy to be hard on yourself when you are not the best at something or make mistakes, but Doudna clarifies that by making mistakes, we are learning to be successful. She really pushes for the importance of learning from your mistakes. I identify with Doudna because in an interview, she addresses a tendency that women often have to underestimate themselves. She is successful in empowering women in science to essentially just go for it, and if you go for it, to give it your all. She also advocates for women in STEM and strongly influences a chain of encouragement for females in science especially. Doudna first graduated with a bachelor's degree in chemistry. She then went on to pursue a doctorate degree in biochemistry at Harvard. She is focused on the structure of RNA and characterization of ribozyme structures, and the investigation of viral RNA. With the help of Emmanuel Carpentier, she discovered the CRISPR-Cas9 and hypothesized that it could be used to edit the genes of living organisms. She is currently teaching molecular and cellular biology at the University of California, Berkeley. Here, she is focused on discovering mechanisms of CRISPR-Cas9 and associated proteins for further gene editing. Doudna's most recent paper is titled Precise Transcript Targeting by CRISPR-CSM Complexes. In this study, the authors began by choosing type 3A CSM complex from Streptococcus thermophilus and verified correct expression in embryonic human kidney cells. Next, the authors used transient transfection, Western blot, and immunofluorescent staining to ensure the correct size, expression, and nuclear localization of the proteins. First, green fluorescent protein, mRNA, were targeted, and the GFP-KD was calculated. Next, they surveyed cRRNA parameters and tested which GFP targeting spacer yields the highest KD. Next, the authors studied the capability to multiplex cRRNAs against a series of targets. These tests proved the variable multiplexing capability of the CSM system. The authors write, "Here we have shown that the type 3A CSM complex from S. thermophilus is a powerful tool for the eukaryotic RNA KD." This quote summarizes the findings of the preceding figure. This figure is a combination of figures that work together to visually portray results from the study conducted in her paper. Figure 1A, cis and trans cleavage of Cas13. Figure 1B shows the locus of S. thermophilus type 3A CRISPR-Cas. Figure 1C shows cRRNA target binding and 6NT cleavage pattern. Figure 1D is a Western blot plot which shows the size and expression of Cas-CSM proteins. Figure 1E, immunofluorescence, which displays nuclear localization as well as expression of Cas-CSM proteins in human embryonic kidney cells. Figure 1F is the relative GFP of fluorescent human embryonic kidney cells, transfected with plasmids demonstrating Cas9, CSM 1 through 5, and the indicated GFP targeting cRRNA. Figure 1G is similar to Figure 1F, but includes CSM mutants. Figure 1H is similar to 1F and 1G, but includes GFP cRRNA altered to specified spacer lengths. Figure 1I is respective GFP and RFP fluorescent of human embryonic kidney cells transfected with plasmids and displaying Cas6 CSM1 through 5 in the indicated cRRNAs. Figure 1J is vectors delivering genetic material promptly to a cell. Figure 1K is another Western blot plot which shows the expression and size of Cas CSM proteins in the human embryonic kidney cells. Figure 1L is the respective GFP fluorescence of human embryonic kidney cells, transfected with delivery vectors and displays specified GFP targeting cRRNAs. Doudna has uncovered the crystal structures of two large RNAs and, most famously, a method of gene editing called CRISPR-Cas9. I hope you enjoyed learning about Jennifer Doudna as much as I do.